Journal Club.
Does Michael know about this?
So, a couple of months ago, Michael decided to start something we call the Journal Club. So basically, we sit down and read a paper that Michael's decided that we will read for the week.
And we've noticed that Chris is typically ill-prepared for these meetings. Word of advice to rising thesis students, the college provides lots of useful programs.
I would be devastated if I were sterile. I mean, it just doesn't seem fair. I'm always so safe in lab and follow all the safety regulations down to the letter. You know, if anyone should be sterile, it should be Dan. Yes, hello, I need to make an appointment. I think I might be sterile. Who do I see about that? Great. Okay, yes. I'll just, at 4 o'clock. Thank you.
If Chris had read beyond the title, he would have seen, to quote, that Microbacterium violaceum is a basidiomycete fungal pathogen that causes anther smut disease of many perennials in the Caraphalaceae family. Infection causes sterility in the plant, and Microbacterium spores take the place of pollen in the flowers.
Yes, hello, what's your cancellation policy?
Okay. The main goals for the presentations are for the students to practice articulating the main goals of their projects in a larger conceptual context, and to also help foster discussion among the students about their projects, their goals, and so on. Is that good?
I didn't know what to do for my presentation, so I'm looking at Michael's presentation for some ideas. So in this lab we study the anther smut disease caused by Microbacterium. Here's a really smutty flower. Here's the disease cycle. First, a pollinator like a bee inoculates a plant with the disease, and then the spores on the plant undergo meiosis, and then the two mating types mate and infect the flower, and then usually the flower overwinters, and then the next season when it blooms again, there's a systemic infection, and it is now a disease. Oh, and here are some flowers. And here's Silene meridema, the plant I'm working with. It's also called the sea campion, probably because it grows by the sea.
You didn't mention anything about hybrid pathogens, Aaron.
Why would I talk about hybrids? They're evolutionary dead ends.
Oh, really? The influenza epidemic of 1918, caused by a hybrid virus? Dutch elm disease that is currently ravaging America's elms, caused by a hybrid fungus? Ligers? Seems like you have a lot to learn about the evolutionary potential of hybrids, Aaron.
Are ligers sterile?
Fact. Ligers are hybrids between lions and tigers. Fact. Ligers are the most ferocious animals on the planet. This is because they have F1 hybrid vigor. The offspring of interspecies matings are often less specialized than their parents, but more fit in generalized environments. i.e., combat. That's why I'm dating an Asian. My kids are going to have F1 hybrid vigor whether they like it or not. Just like Tiger Woods. But even better, like Liger Woods.
Michael's really been encouraging Aaron and Dan to discuss their theses together. I think it's because I still have no idea what Alicia did her thesis on. I know she played around with pipettes a lot, she cursed a lot, but other than that, I have no idea.
So, do you have a project yet?
Actually, I do. I'm going to be working on Silene marittima, and apparently there are two species of Microbotrium, Lagerheimia and Inflate, that are coexisting in St. Patrick, and we're trying to figure out why they're doing that, or if they're even doing that. And yeah, there's the same situation going on with Vulgaris in the Alps, and we have collaborators working on that. So it's really promising and interesting research. What about you?
I'm working on hybrids.
Huh. The competitive exclusion principle. Two species can't coexist for long on a limiting resource. This sort of describes my relationship with Dan. Two pathogens, one host, two thesis students, one lab. Let me put it this way. In the 1940s in Hawaii, the government decided to release 32 species of parasites to combat a pest infestation. Of the 32 species, only 3 species of wasps remained. Of the 3 species, only 1 was victorious. Dan intends to be the Oppius Ophilis of this lab.
What? Who took my last pipette tip?
We all occupy different niches. I basically live at my workstation. Dan has his computer. Michael's always in his office. And I have no idea what Chris does in here. But anyway, it's kind of neat with pathogens. For them, each host is a new niche. And so basically, when you have a whole bunch of different host genotypes, you have a whole bunch of niches for the pathogen. So that might be what's happening with the 2 species of Microbotrium, and why they're able to coexist. Or maybe, I guess we're still trying to figure this out, but maybe we're just stumbling across them in the middle of them excluding each other. But anyway, the real question is, can Dan and I coexist in this lab? Do we have some special relationship where competitive exclusion doesn't apply? Do we occupy different niches? Or do we have to drive each other out?